the next thing you might want to do once you've got an image open is to adjust the visualization parameters. And that's because, as many of you probably saw, the bioformat sometimes will open it at the full dynamic range, which might be 16-bit. So you've got a lot of gray values, and it will appear really dark on your screen. So a useful thing to open is the channels tool window. So if you go image, color, channels tool, this little thing pops up, or some variant of this. You can also do things like split, merge channels, arrange channels. You can do toggling on and off, view them as grayscale, view them as composites. So there's lots of nice little ways you can quickly use this window, and it'll, realistically, I think I open it every time I use FIJI because you're always using it. So yeah. The next thing is there is a tool in FIJI to bring up the histogram. So that's under the Analyze menu, histogram. And this will, for the channel, it'll pop up this little window, and that explains what the minimum, the maximum values are for what's found in your image. This is really useful for setting the display parameters that we're going to go through in a sec. One kind of side thing is if we're merging images together, you really want to make sure that your image source files are not RGB color images. So some microscopes have a color camera, and that's going to be for like histology. You're going to get your pinks and purples and things like that. Now for fluorescence, what we find with those is there's an issue where they're leaky. So basically, the way a color camera works is it's a normal camera with a filter on top where you have red and green and blue filters. Now those filters don't always overlap with your RFP filter on your microscope. And so you might end up with some of your data in the red image being in the wrong channel because it would be picked up, or these filters, the alternate colored filters won't block it. So you should always capture in grayscale. And to prove that point, I have some data. So here's a image from a standard test slide. It's labeled with Alexa 555 wheat germ, I think. And when we do the histogram, it looks all fine. But if we look at the individual channels, there's, mostly in the red. So it's 5 to 199. That's the 199 is the brightest thing in the red channel. But in the green channel, it's 1 to 20. So the brightest thing is 20 pixels, which is about 10% of that. And all the way over in the blue, there's 0 to 5. So it's not as bright in the blue. That's not surprising. But if you start merging these with a channel that has lots of green in it, you might start picking up co-localization because you've already got stuff appearing in the wrong channels, and it'll be vice versa for the green image. It'll have leaky stuff into the red, and then you've got, rubbish data, basically. So you always want to make sure, even if you do capture something like this, convert it to grayscale or 8-bit or whatever it is before you start merging it with other channels. So how do you merge channels? Well, image, color, merge channels. And it pops up this window, and you tell it basically channel 1 should be this image, channel 2 should be this image, et cetera, et cetera. I would normally say, yes, create a composite and ignore the source lookup tables. So sometimes you might have like a green channel that you want to merge into the blue channel in here. So if you click ignore source, it'll apply what the color lookups are labeled here. So yeah, ignore source or keep source images. So if you don't trust that you're going to do it right, you can tick that, and then you get like a 4th window, not you close all 3 and then only end up with 1. So the second exercise is to open the 3 images. I think I've been generous and named them like _red, _green, _blue. Doesn't really matter. In fact, like we don't tend to use red and green because of color blindness. So you could try and even do it with magenta or something if you like. You can save the output, but leave it open because we are going to play with it in the 3rd exercise. So just to save time. So have a go at doing that. Oh. Okay, so most people, everyone has something like this. It's a bit washed out. It's a multi-channel image. You've got the slider down the bottom where it has the 3 channels. We will move on. There's answers on the slides. Okay, so the next thing is you probably look at this image and go, that's nice. My group leader or the publication isn't really going to accept that. So we never want to overexpose the images. We always want to try and underexpose because we can make them brighter, but we can't take an overexposed or saturated image and make it dimmer. So you're probably going to end up with something like this, where you want to bring it up in brightness, you know, in FIJI and not on the microscope. So when we make the adjustments on images, it's important that we make the same adjustments on all images, because you can't just say, oh, look, my control's brighter than my treatment. Hooray, it worked. That's fraud. So there's a number of different ways in FIJI that you can adjust the display settings and that. So it's under the image adjust. Nice and helpful. The main one that most people probably use is the brightness and contrast. And that allows to change the way it's displayed on the screen and therefore on your talks and publications and things like that. There's also the window level that's similar, makes it brighter or dimmer. And then you also have some other menus that will cover at least thresholding later. I don't cover color thresholding anymore. And you can change like the canvas size or something if you need to adjust it, make it bigger. So adjusting the brightness and contrast on the image, you want to, so most people have probably made an image brighter and they probably just dragged the little slider until it looked good. But you probably didn't write in your figure legend, I dragged the slider until it looked good in the paper, because that's not very scientific. It's not very reproducible. In fact, if I got everyone here to make it look good, probably have 50 different numbers. So how do you do it so that it's the same every time and it's reproducible? Well, you use the histogram. So that's why I showed you that before. So in this image, if we do analyze histogram, unfortunately, FIJI says, do you want to do it for the whole image? And you say, no, not for every channel, just the one I'm on. So if you're on the red channel, you run the histogram, it'll tell you that it's something like 4 to 85. And then if you do it for the green channel, it's 9 to 123. And for the blue channel, it's 7 to 74. So this is only one image. In reality, you've probably got control treatment. You've probably got time point 1 through to 50. So if you know that your sequence is going to get brighter or dimmer, pick the brightest one and then set your settings around that. Now, if you have like multiple things that you're comparing, you have to pick one that you use for all of them. But what we're going to do is find these values. And then when we bring up the brightness and contrast window that you're probably all familiar with, and you probably just drag this and slid it somewhere about here and said that'll do. The proper way to do it is to hit set, and that'll bring up a window. So the minimum and maximum, just to cover what I say on the slide, these will make it brighter or dimmer. And as you move these, the brightness contrast ratios change because they're linked. When you change this and you're sliding it around, you're not changing the numbers that each pixel represents. So you can make it brighter and still measure it, and it won't make a difference. It does if you hit apply. So don't hit apply, because that does start changing the data. But when you hit set, oh, hang on, I've got a GIF. So we bring up the brightness and contrast thing. And so, yeah, what most people do are just going to do that and go, that looks about right, and stop somewhere. But what you should do is go analyze histogram. This will pop up, and you look at that min and max value. And then you go set. It's going to bring up another window, and you punch in 4 to 85. And then it's displaying the full display range of that image. So the slide, I've taken out the slide. One cool feature that won't come up again, but you'll see it when you actually, I'll do it on the screen because you should see it. It's quite useful. If you go onto here, and we bring up this set window, there's these two tick boxes. One is propagate to all other 3-channel images. They're really useful because if you've got time point 1 to 50 already open as separate windows, you can just adjust one of them and then set it to all of them and it will work. The other tick box is propagate to the other 2 channels of this image. Not really useful. So, because when you set your brightness for like the DAPI channel, it has no bearing on what you should be setting the brightness to your GFP channel, for example, because you could just add more DAPI or take the DAPI image with a longer exposure and be brighter in that. So they're independent, but each DAPI channel that you're comparing has to be the same, if that makes sense. So have a go at making this picture look better. And hopefully everyone's will look the same. So if you do histogram, it'll have this pop-up that says include all channel or all 3 images, say no, and then it says the red. If you say yes, then you kind of get like a merge of all the different channels and it's kind of useless. So, and one tip, something that I always tend to fall for, if the histogram pop-up is still on top, it will happily give you a histogram of the histogram. So make sure you're re-clicking the image window. All right, so something that came up that I'll highlight is if you have like a window open, if I close this, and if I bring up the histogram of the red channel, probably not that obvious, but see how this is black and this is gray, the text? So what it's doing is it's the histogram window is actually like an image, is what FIJI treats it as. And that's now at the top level, or is what we call the active window. So if I click on display adjust, what I'm doing, you can see this goes to rubbish, is I'm adjusting the brightness of this image that's the histogram. So you want to make sure that this window that you're playing with is the one that's kind of selected. So while this is on top, you can actually change the channel as shown down the bottom there I'm just scrolling my mouse wheel, but this isn't changing or anything. But if I select this, update that, and then as I scroll through, thanks, FIJI, you'll see that it's now like relevant. So just get in the habit of maybe clicking and making that window your main window. That came up for a lot of people. Again, we've got the answers. The key thing is basically don't do it for all channels because that stacked histogram is kind of useless. You can also change the scale of your images. So that's under the image type. So whether it's like, when I say scale, I mean the bit depth, sorry. 8-bit, 16-bit, 32-bit, or change it to RGB and things like that. There's then pretty specific use cases for these. Something just to be aware of though is often if you adjust the display and then you change the bit depth, it will, for whatever reason, use the adjusted settings. So then you are really changing the numbers underneath. So just be careful if you're doing the conversion. There is a edit options conversions, and make sure scale when converting is turned off. But just be aware of that if you are playing with the bit depth.